them in at the end. Now to introduce today's speaker, Michelle Moffat, microbiologist at Western Sydney University. Just pass the screen to you there, Michelle. You should be able to now accept the invitation to present. Now, Michelle's background is in small molecules and how they are important in species interactions, especially pathogen-host interactions. Screen is just coming through there, I think. So here we go. Looks good to me. I think you're probably now ready to get underway, Michelle.
Okay. Well, thanks for inviting me. Okay, so I'll just give a little bit of a background to Myrtle Rust. So Myrtle Rust is a disease which is caused by the obligate biotroph called Austropuccinia psidii. And so it arrived on the Australian East Coast back in 2010. And some of you may have caught the talk a few months ago by Rod Vensham about it really brought home the effect of Myrtle Rust on our local Myrtaceae plants. So Austropuccinia psidii does infect Myrtaceae plants, and so some of those are looking like they might become extinct within the next generation. So this really will impact on the biodiversity of our ecosystems within Australia. It also impacts on Indigenous culture and also on Australian industries. So we want to really investigate how we can sort of overcome some of these issues and identify ways in which we could perhaps restore biodiversity. So as I mentioned, the pathogen is an obligate biotroph, which means that it requires the leaf for its source of nutrients and energy. So what happens is the spores of the fungus land on the surface of a leaf. They germinate and produce these aprosorium, which then penetrate through the epidermis and start to produce haustorium. So this really happens within the first few days of infection, and that early stage of infection is undetectable macroscopically. So we can't see this happening just by looking at the plant. So we don't really see the sort of classic symptoms of Myrtle Rust disease until sort of 10 to 14 days later, which is when we start to see the development of the Uridiospores. And so this is the classic sort of rusty appearance on the surface of the leaves. So detection of this early stage of infection can be quite important. So if we do want to be able to save our biodiversity, we're lucky in one sense that nature has given us a bit of a break, and we see that some of the plants that can become infected with Myrtle Rust show some variability in susceptibility. So there have been a few studies which really delve into this, but one specifically is by Morin in 2012, who studied a number of different taxa and found 11 of those were completely resistant, 27 were completely or highly susceptible. So this is, sorry, I should mention on the left here, this is what the resistant would look like. So no evidence of disease at all. Whereas on the right is highly susceptible. So development of significant numbers of pustules on the surface. But then 83 taxa showed some variability in their susceptibility. So individuals within a taxa could be completely resistant, some could be highly susceptible, and then they're sort of in between. So hypersensitive is really where the leaf is showing some evidence of infection, but some cell death at the site of infection. So can we use this to our advantage? And so one way that we could do that is by really finding out whether we could phenotype for resistant individuals before they're infected. So rather than taking a bunch of plants and spraying them and seeing, oh, is this plant resistant? Could we actually find a way to identify these resistant individuals very early on in the growth of the plant? And so if that was the case, we could take those resistant plants and perhaps use them in restoration of an ecosystem. We could use them to assist Indigenous culture or also assist Australian industries. So that was one real driver for this project. We also wanted to understand perhaps what are the differences in these different phenotypes during early infection. So that stage where I mentioned at the beginning, where the disease is not visible, but is really at the crucial stage of infection. So what's actually happening inside the plant during those early stages. So one study that's looked at what the differences are in these resistant, hypersensitive and susceptible phenotypes is sort of a microscopic evaluation. So this study was done on eucalyptus plants, and it was evident in those plants that within the first 48 hours, resistant plants are able to halt the infection process by preventing penetration of the acrosorium into the plant epidermis. So that happens within the first 48 hours on eucalyptus plants. Hypersensitive plants seem to have some penetration into the leaf epidermis, but at that stage, there's some pathogen recognition by the plant, and that induces host cell death. And so that in that stage is where we see prevention of further infection or continuation of the Austropuccinia psidii life cycle. And then in our susceptible plants, the life cycle of the Myrtle Rust is able to continue all the way through to the end, where you get the development of uridinia pustules on the surface of the leaf. And so in these eucalyptus plants, this is happening over around 10 days, but this time can vary depending on the species. So that's a microscopic view of what's happening, at least in eucalyptus. So how can we sort of understand what's happening in more detail inside the plant host? So there are a number of different ways that we can look at resistance and understanding what's happening during early stages of infection. So one thing that we can look at is genomics. So we can compare the genomics of the plant host. And so there have been some studies which look at genome-wide association studies to really identify, you know, if we wanted to identify resistant plants, this could be one tool that could be used and has been investigated. So once we have the genome, so in a cell, when or a plant host is responding to stimuli, it does so by switching genes on and off. So if we can look at the genes that are being switched on or the genes that are being switched off, either as a result of resistance or during early infection, we can understand what's going on inside the plant. And so there are quite a number of studies now that have investigated the transcriptomics in different plant hosts at different stages of infection. And so this is starting to help us understand how the plants are responding to infection, but also how they might be able to resist the disease. So once genes are switched on, it's not all that straightforward, because once the genes are switched on, the plant cells are still very good at finely tuning their response to the stimuli. So there are other ways in which these genes that are switched on can be further regulated. And so obviously genes are translated into proteins. And so we could look at the abundance of proteins inside the cell to try to understand, well, what are the proteins doing and how might they be important in resistance or early infection. And so there was a study by Sakya looking at Eucalyptus grandis and looked at the proteomics at that level. So we're still developing this understanding of what's happening at the protein level. So what is the purpose of proteins? Proteins are often there to catalyze reactions. And so we can then look at what are those proteins doing to understand how they might be important in resistance or early infection. And so this is where we get to metabolomics. Metabolomics is the investigation of small molecules within a cell. And so we can look at changes in abundance of those molecules between plants or during early infection. So this is now being investigated in Eucalyptus grandis, and in this study, which I'll be talking about today, in which we looked at Melaleuca quinquenervia. So yeah, all of these different tools are fantastic because they all work together and they help us to sort of understand in more detail what's happening. And we can, the more we know, the better we can understand the system. So in particular, we used a tool called Untargeted Metabolomics. So kind of like with genomics, genomics is the study of all of the genes within a host genome. We could, if we might know that we were interested in one particular gene, we could target that gene, but we often get more information by looking at the whole genome. And so untargeted metabolomics is kind of like genomics. So rather than just simply looking at one molecule or one group of molecules that we think might be important in resistance, here we're looking at basically as much of the metabolome as we can. So we're looking at a broad range of molecules at a particular time. Usually we don't know much about what those metabolites are, but it can give us a broad sense of what's being kind of changed in abundance over time or in response to a stimulus. So this is the process that we use for untargeted metabolomics. We take a sample of a juvenile leaf that would normally be susceptible to Myrtle Rust infection. We place that in a solvent with some beads and bash it up with a bead beater and sonicator. And so that releases all of the soluble material from inside the leaf cells. And so you end up with this nice green solvent mixture which contains all of the metabolites. We then run that through a mass spectrometer, which analyzes the mass, but also some of the characteristics of each of those metabolites. And then we use data analysis to analyze that output. So untargeted metabolomics is actually gaining a lot of traction at the moment. Basically, we can use it to gather what we call biomarkers. So these are just basically markers that suggest that a disease might be present or resistance might be there. So it's really being pursued heavily for looking at human diseases. So an analogy might be if you're going to the doctor for a checkup and they take a blood test, they might look for biomarkers that are indicative of something like liver disease. And so if those biomarkers are there, then they could say, well, you have liver disease, we need to do something, do further testing or something like that. So what we want to do is be able to give our plants a health check to tell us, is the plant going to be resistant to Myrtle Rust disease? Or maybe the biomarkers could tell us that the plant is in an early stage of infection before we can actually see signs that it is infected. As I mentioned, metabolomics has already been used quite, or it's starting to be used more frequently. And so there are other studies looking at other diseases, resistance to other diseases, as well as resistance to abiotic stresses as well, such as drought. Okay. So I'm going to be talking today about the study that was published earlier this year. So we were investigating Melaleuca quinquenervia, which is a keystone species in some of the swampy ecosystems of northern New South Wales. So these ecosystems are being sort of in a hot spot really for Myrtle Rust on the Australian east coast. So there is really a strong variation in the susceptibility of individuals to Myrtle Rust. So with the help of Louise Shuey and Jeff Pegg, we took samples of resistant individuals, hypersensitive individuals, and susceptible individuals. And we analysed them over time. So before infection, 1 day, 2 days, and 5 days after infection. So really in that early critical stage of early infection. For those of you who are interested in finer details, we did some solvent extractions with methanol, acetonitrile, and water mix. We have a water system here at Western Sydney University, so those metabolites were run on a UPL system with a high definition mass spectrometer with an ion mobility. And so this system is really nice because it sort of gives us a lot of detail about differences between different molecules. So what did we find? Basically, when we looked at our samples, we identified approximately 11,000 different well, we call them molecular features. Some of them might be related. So it was about 11,000 molecular features. We then looked at how their abundance changed during the experiment. We found about 2,000 of those changed according to time. So that was the most prominent effect, was time. We saw that just less than 1,000 were changed in abundance according to their phenotype, so susceptible, resistant, or hypersensitive, and the remaining were changed according to the interaction. So really what we saw was that time was the most influential factor which drove the metabolites in our samples. So this is a heat map. My husband calls it the Hulk map because it has the Hulk colors. Don't know why, but we'll go with it. So if you haven't seen a heat map like this before, each of the columns represents an individual sample, and each of the rows across here represents a different molecular feature or metabolite. So they're grouped along the top here. Each of the individuals is grouped according to how similar it is to other samples. So there we see that the samples really grouped together according to the time. So this is the time 0 before infection, 24 hours later, 48 hours, and 5 days. Now, all of the phenotypes are indicated here by these colors. So hypersensitive is dark blue, sensitive is light blue, and resistant is green. And so they didn't really group together according to phenotype. So here we see that time was really the driver in this heat map. So there was a strong metabolic defense response, I guess, during this early stage of infection. So a fair chunk of these metabolites, basically what this is showing is that these metabolites were switched on or increased in abundance, particularly after 24 hours, and then still there after 2 days, but then returning back to pre-infection levels by day 5. So what this tells us is that regardless of the phenotype, those metabolites are being switched on. Okay, so the plant is recognizing the Myrtle Rust germinating on its plant leaf surface, and it's responding in a strong defense manner. So I mentioned earlier about biomarkers. So we could, if there was a need, we could develop this metabolomics tool as a way to detect early infection in the first 48 hours. So we could look for any number of these metabolites in a sample of a leaf, and that would tell us if the plant was infected. And that would be regardless of whether it was resistant or susceptible. So can we understand from that what the plant is doing when it's sensing Myrtle Rust infection is imminent? So unlike genomics, which is fairly far along in being able to predict functions of genes, I know genomics is not perfect, but we can do a pretty good job of predicting functions of genes. Metabolomics is not quite at the same level, so we can still get a fair appreciation of what some of the metabolites are. We can predict them based on mass, fragmentation patterns, ion mobility patterns. But we can't be 100% sure unless we do follow-up experiments. But we can sort of get a fair idea of the class of molecule that these metabolites belong to. And so we saw that infection really triggered enrichment of fatty acids, dipeptides, and triterpenoids. So terpenes are interesting. Myrtaceae are known for their terpenes, and so there have already been some studies that have investigated the role of terpenes in Myrtle Rust infection. So here we're seeing that they're increasing just in response to infection, and it doesn't really have any bearing on the phenotype. Terpenes have also been associated with other biotrophic pathogens in other plant hosts. So we know that they're really important in responding to pathogens. But it may just be that different terpenes have different roles. So if we really wanted to understand what these terpenes were doing, we'd have to really investigate more thoroughly what the structure of these terpenes were. We could then match it to transcriptomic results and other things. So really, this is just a teaser, I guess, and really a lot more work has to be done to figure out what these terpenes are doing in response to infection. So did we see a difference in phenotype during early infection? Well, not a massive difference. So there were only really 19 features that were unique to the resistant phenotype. So, here I was telling you that we identified 11,000 in total, so 19 is not a very, large number. So there's not a big difference in the resistant phenotype during early infection. But of those 19, some of them were defense-related fatty acyl glycosides and hormone-related metabolites. So things like phytohormone defense pathways linked to salicylic acid biosynthesis and signaling. So it is worthwhile to also investigate some of these metabolites further. They may have some important role, even though there's not many. So I mentioned at the beginning that we were also really interested in understanding if we could develop metabolomics as a tool to identify resistant plants before they've been infected. And so this is a heat map just showing the day 0 time point, so before infection had occurred. And so we can see that the individual plant so each individual is grouping together according to its phenotype. And so we can see here these ones on the right, these are the resistant individuals. Okay, so they are showing that they're a bit different than the hypersensitive and the susceptible individuals. So what that tells us is that there is a core set of metabolites that are different in their abundance. So some are lower in abundance and some are higher in abundance before infection is actually occurring. So I guess this is really, since perhaps during resistance, the Myrtle Rust is not fully penetrating the plant leaf, then perhaps some of these metabolites are playing some important role in preventing that process. What are some of these molecules? So again, we have a pretty big group of metabolites here. We can't characterize or we can't predict all of them, but of the few that we were able to predict, some of those belong to secondary metabolites, including flavonoids. So flavonoids are, this is an example of a flavonoid here. If you've been told to drink red wine because it's good for you, that's because of the flavonoids. But flavonoids can also have bioactive properties, and so could be responsible for inhibiting germination of fungi. They can have antibacterial effects, and they have other roles as well in the plant. So really, these flavonoids could be something to investigate again into the future. So was it just a fluke that we found these core sets of metabolites that were increased in Melaleuca quinquenervia? We wanted to investigate. This is not in the paper that was published this year, but this is a follow-on experiment looking to see whether this result could be identified in other susceptible and resistant individuals of other taxa. So we looked at Eucalyptus grandis. Here we only compared resistant here on the right and susceptible on the left. But again, we see that metabolomics can do a fairly good job of differentiating between the two phenotypes. So that indicates to us that it's really worth looking at this technique in more detail. So outcomes of the project were really that metabolomics can be developed into a screening tool for resistant plants, as well as a tool for detecting plants during their early stage of infection. So that was a nice result. So we're really now continuing that work from the E. grandis to see, well, could we take a bunch of these unknown Myrtaceae plants of different taxa to see, okay, if we snip off a leaf, do we see the same pattern of metabolites being changed in abundance according to their phenotype? And then if we do identify our resistant plants, these could then be used for, breeding programs. They could be planted out for restoration and so on. So watch this space, fingers crossed that we get some good results with that. Also really keen to understand in more detail the resistant mechanisms associated with some of those key metabolites that we identified, particularly the flavonoids. I'm interested in secondary metabolites, so that's a win for me. So that's the end of the talk. Really, this project would not have got off the ground if it wasn't for the amazing support of loads of different people, particularly Jonathan Plett at Western Sydney University, Perry Tobias and Robert Park at Sydney University, and Louise Shuey and Jeff Pegg at DAF in Queensland. And then the two that really conducted a lot of the research were Johanna and Donovan from Western Sydney. And so the research was supported by the Australian Plant Biosecurity Science Foundation. And so again, this wouldn't have been possible without them. So really excited that they were able to support the research. So that's it. Thanks for listening in. Oh, I can't hear. Why can't I hear?
I can't hear you either, Renee. That was an excellent talk, Michelle. That I thought it was really interesting. And can you hear me?
Yeah, I can
I
hear
can hear you, Marge. Can you hear me?
Yes, we can hear you now, Renee.
Why the heck have I lost my webcam? Oh my goodness.
Not sure.
All right. Well, you know what? I really thought that was a fantastic talk, Michelle.
Thank you.
I can tell that you have had a great deal of experience teaching, because you certainly made a complex subject quite clear. Now, we don't have any questions in yet. I'm back. I'm back. Fantastic. I'm back, and I've got a stripy face, which is also interesting. There's not much I can do about that right now, but at least we seem to have ameliorated the worst of the technical carnage, and Marge, of course, is standing there, standing by in case it returns. So we do not have any questions in the question box yet, so please do get them in, audience members, but I do have some of my own. So it was great to hear you mention at the end there you're going to move on to look at different species, because of course we just have so far seen some results from within one species.
Yes.
So it's an obvious follow-up point really, isn't it?
Yeah, that's it. Yeah, so I guess it's just about seeing, I mean, the Myrtaceae are so varied. And so I think, you know, if we can really tap into this ability to identify resistant plants, then that could be really important for future work in being able to restore some of these ecosystems. So unfortunately, not all taxa have this variation in susceptibility. So I don't know what we can do in that case, perhaps some specialized breeding or something to try to stimulate some resistance, but at least for those that have variation in susceptibility, this is a really good tool.
I think so, and of course you mentioned a number of tools there available to plant pathologists, starting right back with genomics, moving through transcriptomics to proteomics to metabolomics, and I'm really not, from my own background, terribly familiar with the proteomics and metabolomics approaches and the challenges inherent within them, but I suspect, you know, we have a spectrum of expense and technical difficulty, and actually metabolomics is probably one of the more accessible, if most accessible, of that set of tools, especially looking at the method that you displayed there.
Oh yeah, it's, you know, we've done some transcriptomics as well, and you know, you have to be so careful with the RNA, and you know, these it's super straightforward. You just, you know, chuck the leaf in some solvent, beat it, and whack it on the mass spec, and it's, you know, there's no care in the sort of RNA scheme of things. The data processing is a little bit more in depth, and it depends on the tools you have. But you know, I think that over time that will really improve. Yeah, so we are also sort of looking at how stable those metabolites are. So whether we could take the method out into the field, just grab a leaf from a field sample, and then bring it back to the lab without sort of that worry of having liquid nitrogen or, you know, dry ice or something. So if those metabolites do seem to be fairly stable over sort of, you know, a couple of days, that could really improve things as well, because we, you know, that could be helpful for, yeah, field sampling and things like that.
So perhaps one takeaway message for researchers who perhaps aren't so familiar with this technology and approaches: do not be afraid.
Yes.
Perhaps more accessible than you might think, especially if one is able to team up with an expert such as yourself, of course.
Exactly. So I'm always happy for collaborations too. So just
Very
give a
good.
shout.
Yeah, because one thing I do know from my own experience is some of the technical difficulties we can have with the transcriptomic and next-gen sequencing type approaches is that you can have just not a great run, or for some reason you're suffering from a lack of sequencing depth. And so when you mention there's only 19 unique features, I get the feeling we can probably trust that data, and it's not just about perhaps a bad performance of that particular metabolomics attempt.
Yeah, exactly. Yeah, I guess one thing with metabolomics is that we have different mass specs and different ways that mass specs can run. So you could get variation across instruments. So you might have to develop your method specifically for the instrument that you have. Whereas with next-gen sequencing, I think there are different tools that you can use, but it's a little bit more standardized. But, once you've got that out of the way, I think it's fairly straightforward.
Very good. We do have a couple of questions come in now from the audience. Great talk there, Michelle. Do you have any information on the frequency of resistant and hypersensitive phenotypes in M. quinquenervia in the wild?
That's a good question. Jeff Pegg is and Louise are really they're the ones that know that stuff in and out. And Jeff made the joke the other day that I don't even know what plants look like. So when you're working at the molecular level, yeah. But I don't know in the wild, but I know that, and I've heard this mentioned a few times, that in the wild, sometimes when the resistant plants get hammered by myrtle rust year after year, and particularly after the bushfires that we had, some of those resistant plants seem to lose their resistance, which is really a scary thought. So it would be really cool actually to do kind of a study in that sense, to look at how those plants are changing as they're getting hammered. Yeah, because that's kind of a scary thought.
Depressing in news for me, difficult to study though, right? Because
yes,
how are you going to replicate that in a controlled setting?
Yeah, I don't think the uni would be too impressed if I had some plants and set them on fire. And it would have to be sort of, getting out into the field again and seeing how that could be replicated maybe.
Yes, tricky. And we do have one more question here. Another thanks for a fantastic talk, and apologies for missing the very start. Hopefully you didn't already address this, but I don't think you did in any detail. How were the phenotypes determined for the experiment?
So that was done by Louise and Jeff. So they had already identified the phenotype before the study. So they spray or infect the plants before. So these are all just greenhouse plants. Once they are sure of the phenotype, they cut them back to generate new growth, and that's what we used for our samples. Yeah.
All right. Very good. I'm just going to get you to speak a little more to one of my questions before I make my way through one that's just popped in the box there. I was just wondering, I need to speak to this a little bit, to what extent one can go backwards. So we understand here we have a metabolite, therefore this would be the protein, therefore this would be the transcriptome, and therefore this would be the gene.
Good question. Well, I guess first of all, we would have to sort of really be confident of the structure of the metabolite. So that process really involves isolating the metabolite itself, doing some chemical characterization, so things like NMR is the tool that's commonly used for structural characterization. So once we know really what the structure is, you can use some sort of, I guess, investigative work to see how those kinds of molecules are made in these plants. So, you know, terpenes are made by terpene cyclases, and flavonoids are made by polyketide synthases. So you could look at how these, you know, what is known about how these molecules are made. You would look for those proteins or transcripts in your data set at the same time point. There are other tools that you can use. So there is sort of this emergence of this kind of networking tools that really look at which transcripts are being increased in abundance at the same time as metabolites, and they sort of use this networking. So that's also a possibility as well. So yeah, a lot of work. Yeah.
I can imagine an entire PhD perhaps just tracking one metabolite going backwards.
Exactly.
All right, so we have a question here that's a little bit different, and perhaps speaking to the increasing recognition of the value of science working in with Indigenous peoples and cultures and beliefs. I am an Indigenous artist. I was wondering if you'd be interested in testing once Indigenous mana whenua karakia or prayer has been spoken with the trees and the plants that the sample is taken from. So of course that's something that we're really just getting underway with here in New Zealand, and what might be easier for you to comment on is to what extent you're working with Indigenous people or your university in general is, or what you might like to do in the future.
Yeah, so we didn't work specifically with Indigenous peoples for this project. Kind of as an aside, we have just started developing a project which is completely different to this one here at Western Sydney, to see how we could use Indigenous artists to, I guess, produce a sort of express how Myrtle Rust impacts on their culture. And so we wanted to use it as a way to kind of broaden, I guess, the understanding of Myrtle Rust in the broader community. So sort of as a way to sort of increase people's recognition of the disease and how important it is, not only to our ecosystems, but also to our Indigenous peoples. So that's really a completely different project to this one. But yeah, I'm more than happy that, you know, if we could, you know, focus particularly on species that are important to Indigenous cultures, that would be something that I'd be really keen to look forward to in the future as well. But yeah, nothing at this stage.
Excellent. Well, we have one more question in the box, and I'll probably wrap it up there after that. Another tricky one really, which is almost more about kind of operations and future research, and you can probably only answer this in the context of Australia, but is there any advice for those in Australia at the moment in the field to be on the lookout for potential resistant plants? The current advice is to cut out anything and not really look around for the resistant plants. Do you know if there's anyone actively looking for resistant plants in the field, that in Australia at least, that people perhaps could get in touch with?
So I know that Jeff and Louise do some surveying in their area, and Jeff is working with some Indigenous groups on Ngari, which is an island off the coast of, I guess, southern Queensland or northern New South Wales. So they have been looking at resistant plants. So they really do some good field surveys of where the resistant plants are, which ones are susceptible. So they're really the go-to people for that kind of work. But yeah, once we establish whether this tool could be used in the field, you know, I really would think it would be worthwhile to take more samples from the field and investigate resistance of those plants.
Yes, I think so. And unfortunately, I can't really offer advice for New Zealand. Things are a little ad hoc at the moment in terms of screening for resistant plants out in the field, but hopefully in the next tranche of research down here in the next few years, perhaps something more systematic and nationwide and focused on that might get underway. All right, I think I can wrap it up there. We've had quite a long session this morning, but it's fantastic to see how much of the audience has stuck with us as we head into midday and the lunch hour. So thanks again, Michelle, for that excellent presentation.
Thank you.
No worries. It's been great to have you, and a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and it will also be emailed to everybody who registered for this particular webinar. Now, the next webinar will be on Wednesday, June 8th, and we will hear from Robert Barreto. He is a professor at the University of Viçosa in Brazil, and he will be talking about hyperparasites in his talk titled Fungal Antagonists of Myrtle Rust from Brazil, Diversity and Biocontrol Potential. So look out for that invite in a couple of weeks' time. And that's it for today, everybody. Haere rā. See you next time.